Now, our second group we're going to be discussing is what is known as the lipids. Lipids, fats, non-polar hydrocarbons. Remember, non-polar means that there's no unequal sharing anywhere of electrons. So there's no positive-ish, negative-ish areas. Because they are non-polar, means they do not play well with water. Thus, they are hydrophobic. Now, lipids, what's great about them? Well, they have multiple functions. They're great long-term storage. The adipose, for those of us who are a little bigger than we should be, that adipose we have. Those are cells that have stored a large amounts of lipids. It's great for long-term storage in us animals. You get enough lipids, enough fats surrounding your body, underneath the skin, underneath the fur. It's great for insulation. Think about it. Bears, many things that, like the otter to the left there. They use fat as a means of insulation. Keep their core body temperatures up when they're in freezing sub-zero environments. Later chapter, when we're talking metabolism, we're going to see that it took energy to make the lipids, to store them away, so that when you go and you start breaking them down, you're going to get some of that energy back. Many of the lipids, since they're long chains of carbons, can be used to make hormones. Turns out, testosterone, estrogen, many of the hormones like those are relatively the same. They're just long chains of carbons that have been bent around to form geometric shapes. Difference between estrogen and testosterone, a couple of functional groups. Same we're going to see with many of the hormones like cholesterol, testosterone, estrogen. And the lipids, most importantly, are going to be what are used to make membranes. As we're going to see in a later chapter, membranes act as the boundary, inside, outside, outside, inside of the cell. Keep stuff in, keep stuff out. Now, lipids. Lipids are the catch-all. Fats, oils, waxes, phospholipids, steroids. All of these fall under the quote-unquote lipid category. Now, unfortunately, our little monomer, polymer that we were talking about a previous chapter, the Lego building blocks, doesn't work here. Lipids, they're too different. What we're going to find primarily with a lot of the lipids, instead of, a couple of monomers coming together to form a polymer, what we're going to see here is a couple of different components coming together. These components, glycerol and fatty acids. Glycerol, a 3-carbon chain. Fatty acids, long chains of carbons. 1, 2, 3 dozen carbons. And their accompanying hydrogens, all linked together. Fatty acids will join to the glycerol through what is referred to as an ester linkage. Here we got an example. Glycerol, 1, 2, 3 carbons. Fatty acid. The artist here is showing you, here's the hydroxyl, here's the hydrogen that is going to be pulled away. Dehydration reaction to form this weird carbon-oxygen-carbon double bond oxygen. This right there is what is referred to as an ester linkage. And this ester linkage is found only in lipids. Triacylglycerol, which we're looking at right here in this bottom box. Tri meaning 3. Acyl has to deals with the lipid, the fatty acid. The glycerol up here what they call the head. 1, 2, 3 carbons, 1, 2, 3 fatty acid chains. Notice it's nothing more than, once you get past the ester linkage, carbon, 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 and a bunch of hydrogens. Triacyl glycerol. Now, saturated versus unsaturated comes back into play here, because whether or not it's straight or may have a kink to it will impact the overall shape of our lipid. Stearic acid, which we're looking at up here, is a saturated fatty acid. That means every carbon has at least 2 hydrogens. The one at the end has 3. There are no carbon-carbon double bonds. What this allows for is that the lipids that are made with stearic acid or other fatty acids that are saturated, they can pack in closely. So when they're in membranes or they're in fats, they pack in tight, which means their structure, their function allows that at room temperature, these are going to be somewhat solid. Think butter. Think the fat you're going to find in animals. Whereas unsaturated, right here, we have a carbon-carbon double bond. What that means is that instead of straight like the artist has drawn it here, it's going to be more like this. A slight bend, maybe depending upon what else is going around, it could be even a more severe bend. How many carbon-carbon double bonds? Mono, poly, single bond, double bond, multiple double bonds means even more of a kink. By having this kink means that the phospholipids or the triacylglycerols or whatever cannot pack in tight. It means they're going to be very loose. They're going to be pushing away from each other. I like to use the example of saturated. Saturated, think about a whole big group of people trying to fit through the door, and they're all packing in there as they're trying to push their way in. They're shoulder to shoulder, straight. Whereas if 1 or 2 people start shoving their arm out, pushing people away, start spacing out. That shoving your arm out means you're bent, means you're an unsaturated fatty acid, means these are going to be more movable. Think more along the lines of oils, vegetable oil, olive oils. Now remember, carbon-carbon bond is 2 inverted tetrahedrons, unless it's a double bond. Then that's a flat plane. That flat plane can induce the kink depending upon whether or not it's cis or trans. If it's cis, you see the 2 hydrogens are both above the plane of the double bond, meaning they're going to give it a kink. Means that these fats are going to be more liquid. Whereas if it's trans, even if it's a double bond, trans fatty acids, things like that, means there's no kink. Means they're going to pack in just as tightly as the saturated fatty acids. Another type of fatty acids are what are known as the waxes. Waxes are found all over in nature. Beeswax, every plant makes wax. It's called the cuticle. Go next time you're outside, grab a leaf that's still nice and green and gently scrape your thumb across the top of it. A waxy white substance will scrape off. That's the cuticle. Since it's a wax, it's a lipid, it means it is very hydrophobic. It allows for plants to keep moisture in. Bees, insects use it in making some of their hives because it keeps water out, keeps water in, allows them to maintain a specific environment. You're also going to find some wax or something similar to waxes on feathers. That's why ducks and geese and things like that can get in the water and they never actually get wet, because their feathers are going to be covered in oils and waxes. From a biological standpoint, to me as a cellular biologist, one of the more important types of lipids are going to be what are known as phospholipids. Phospholipid. They misspelled it. It's supposed to be an O there. Phospholipid. Oh, there it is. So, sorry, chemo brain. Phospholipids. Rule of thumb in biology. If you don't know a word, break it down into its parts. Phospholipid. So, let's break that down. Phospho means there's a phosphate group. POO4. Phosphate. Hey, another one of those functional groups talked about previously. Phosphate and a lipid. There's our 3-carbon glycerol. Here's 2 fatty acid chains. Now, if you think back to the previous chapter talking about functional groups, talked about the fact that phosphate gives up its some hydrogens off the hydroxyls, and it can be charged. If it has a charge, that means it likes to interact with water. But here we have hydrocarbons. Hydrocarbons do not like water. So you end up with a hydrophilic portion and a hydrophobic portion. Excuse me. Blue ball, phosphate. Black, carbons. Red, oxygens. So, you have the phosphate. I'm sorry, not blue's not the phosphate. This pink right there is the phosphate. 1, 2, 3. And there's a 4th one hidden behind it. Oxygens. Going down to our 3-carbon glycerol, 2 fatty acid chains. Now notice this one has a bend in it, means it's unsaturated because right there, carbon-carbon double bond. If they were both straight, that means these phospholipids could pack in very tightly. But because of this little bit here sticking out, means they're not going to be able to pack in tightly, means this phospholipid is going to be able to move around when it's grouped together with other phospholipids. And it's going to be grouped because you have a portion that is water-loving and you have a portion that is water-hating. We call this amphipathic. Great thing about phospholipids, you take a bunch of them that have been dried out crystals, you dump it in water, they're immediately going to form a bilayer. What we biologists also think of as a membrane. There's going to be water out here, no water there. It's a natural state of matter. No energy to make this happen. They're just going to do it. And we'll talk more about that in a later chapter when we talk membranes. We also see that our phospholipids can form steroids, form hormones, cholesterol, cortisol. Each of these geometric shapes you see here, right there, there, and there, each of these is carbons. Every vertice, every corner, right there, right there, right there, there. Each of those is a carbon. Long chains of carbons. This double line here means there's a double bond there. And if nothing's showing, just assume there's 2 hydrogens pointing out. Notice the difference between cholesterol and cortisol. Just what's attached. If you were to remove some of these and add in a methyl hydroxyl there, remove all of this, you'd have testosterone. Add in a little here, a little there, you'd have estrogen. They're all based on these long hydrocarbons that don't like water. We find that many of these are going to be synthesized in the liver, going to be synthesized in, spleen. You take cholesterol, you cut parts off, you twist, you tweak, you cut, you paste, you get our hormones as testosterone, estradiol. One of the reasons why you need vitamin D, whether you drink it in your milk or you get it in your vegetables or whatever, you take a supplement, it's because the vitamin D acts as the base molecule to begin assembling these large geometric shapes.